in the afternoon. So without delay, it's my great pleasure to invite the second speaker, Dr. Boris Parent from INRA, LEPSE, France. His talk will talk about genetic variability of plant response to evaporative demand and water deficit with focus on the phenotyping to the field modeling and issue time. Please.
Yeah, thanks, and good morning, everybody. Thanks to the organizer for, sorry, I will speak only in one microphone. Many thanks to the organizer for the invitation. Really an honor to be here and to give this talk in front of you. So your huge assembly, it's quite impressive. So I will talk about what we are doing in the LEPSE laboratory in Montpellier, in the team of François Tardieu, and how we analyze the genetic variability of plant responses to the environment, so to soil water deficit and the evaporative demand, and how we try to simulate, to study the impact of this genetic variability on plant performances in the field. So as an introduction, you've seen in many talks that we like to describe the impact of genotype and the environment with this kind of equation. So a trait can be described as the effect of the genotype by itself, the genotype being either a line, a cultivar, or a genetic identity like a QTL or a set of different QTLs. The environment and the interaction between genotype and the environment. So two examples. The first one is on yield for several lines of wheat. So here in this graph, you have the relative advantage of 4 lines compared to a Czech cultivar in different environments in Australia. And what we can tell about that is, first, if you look at this graph, we can say it's a mess. Perhaps there are some tendencies with perhaps, for example, the blue genotype gladius being a bit above the other ones. But there's really a huge deviation from this tendency. And this deviation in, there are the interaction between the effect of genotype and the environment. And another example is this time with the genotype being defined as an allele at a QTL. So here it's the effect of this allele in a real population in different environments in Australia and Mexico. So what we can say about that, if we take the environments, the Mexican trial, this allele has more or less always a positive effect, and even sometimes a huge effect of 15% on yield. If you take another set of environments in Australia, in a well-controlled environment with additional irrigation, this allele can have a negative effect. And in other trial in Australia, there's more or less no effect. So here, again, the QTL by itself has more or less no effect, but it's really what we can observe is the interaction between this QTL and the environment. So now that's raised the question, for a given trait value for an effect of allele, what will be the effect in different scenario in the field? So we really have then, that's the objective, to study the link between these traits in multiple scenarios in the field and the effects of a genotype and an allele, and that is for many environments. So the first strategy, which is used, is to measure a trait like yield in many environments in the field, and with a good record of the environmental condition, is to try to define robust effects of the alleles or robust effect of genotype. So how I will give you examples that can be quite common in multi-environment genetic analysis. But really often the environment is defined as a factor, as for example the name of the trial, and not with the environment being defined as quantitative variables like temperature, saltwater potential, etc. And so in this case, prediction, so going from allele to the trait, is not really straightforward. So a few examples. So there's a good review on that, a paper of Franz Vadelsvik in 2011. So the first example, they found a good relationship between the effect of an allele at QTL and the minimum temperature in a heading stage. So with this kind of relationship, we can imagine that in a new trial, in a new condition, with a new environmental scenario, this relationship will be still valid, and though we can imagine the impact of this allele in this new environment. So we'll have other example tomorrow, for example, in the talk of Delphine Fleury, and so that's for in wheat, yield and yield components, and the environment was well defined in for 11 environment, and we have found a robust relationship between allele effects and environmental conditions. Another example, two examples you have in the talk of Émilie Millet. So she analyzed yield components in 60 environments with maize, with a really well-defined environmental condition, and she found also really robust relationship between the effect of allele and environmental condition. So here again, we can imagine that if this relationship is robust in a new environment, we can guess that it will be the case, and we can imagine what will be the effect of this allele. So today I really speak about the other strategy that I call forward strategy, is coming from a proper effect of the allele and use modeling strategy using crop models to simulate what would be the effect of this allele or this trait in multiple scenarios. So I define a bit better this strategy. So we start with hundreds of genotypes, and phenotyping platform. We can measure different variable, different traits with ecophysiological models, and then define a phenotypic profile. That's a set of biological traits for each genotype, and these traits have to be independent from the environment. This trait, we will insert them in crop model to simulate what will be their effect. It means that this trait or variable have to be also parameters of a crop model. That means that we really need the parallelism between the models that we use for phenotyping and the small modules, small pieces of model that are inserted into the crop model. And then if we have this crop model and the phenotypic profile, we can simulate in different climates, in different environments. And then in this case, the output, for example, yield in different environments, from this output, we will simulate then the G by E interaction. And this G by E interaction is then the emergent property of the model and not something that we have measured. And of course, we can use also other field network to validate the approach. If we have performed genetic analysis on these genotypes, so in panels of genotypes, it means that we can calculate the phenotypic value from any composition of allele, and we can get the phenotypic profile for new genotypes or virtual genotypes. And again, perform the simulation. So that should answer the question for a given trait value, an effect of an allele, etc., what will be the consequence in many, many different environmental scenarios. So there are only few examples in the literature. There are more kind of proof of concept. One which have passed the proof of concept is to try to simulate heading date in bread wheat from ecophysiological model and parameter estimated with QTLs. That's more, that's a bit older, but that was also a proof of concept. So in the paper of Yin et al., he tried to simulate yield with the crop model and with parameter estimated by with QTLs. So the result was not really, really good, but at least he found that this model was not worse than the model with estimated parameters. And the last example, also a proof of concept in the paper of Chenu, 2009, with the measurement of leaf expansion rate and the responses to soil water deficit, they found QTL, inserted that in AppSyn, performed hundreds of simulations in Australia, and found where and when an allele had a positive or negative effect. So I start with the modeling and phenotyping part. So we've done phenotyping and genetics for plant growth and efficiencies under drought. So growth, expansive growth, radiation use efficiency, and light interception. And then I will give a few examples on how we can study the impact of the genetic variability on plant performances. For expansive growth, we use a phenotyping platform, Phenodine. In this platform, we can measure 600 plants at the same time. We have sensor for leaf expansion. We have balances for measuring transpiration rate and soil water potential, and a set of environmental sensors. Then we can follow the time courses of environmental condition and transpiration, soil water potential, and leaf expansion at the time step of minutes, from 3 minutes to 15 minutes. So the strategy is for modeling and phenotyping leaf elongation is to start with a model of development. A model of development will give for any leaf rank, sorry, the pointer is not working, no, I will do without. For any leaf rank, the timing of leaf initiation, the beginning of leaf expansion, leaf appearance, and the end of leaf expansion. Then to use and develop a model for the maximum leaf expansion rate, so for any leaf rank, and also the responses to the environment. So the idea for all of this model is to have a really low number of parameters, and these parameters have to be easily measurable in our phenotyping platform or in the field. So for the model of development, for example, that's the only example on the parameter. We have only now this model valid for many hybrids with only 5 parameters, like the final leaf number, the phylochrone, and some thermal time for one or few leaves for the big leaf appearance and appearance of the ligule. For the maximum leaf expansion rate, so we can measure that, of course, with the platform. So the leaf expansion is maximum during the night. So that's the leaf expansion rate for successive leaves in different genotypes in 5 species. So the black, the leaf 3, and then 4, 5, 6, 7, etc. So we use that to get simple models. So for example, increase in maximum leaf expansion. So that's, we can use that in different genotypes or species. Sometimes it's simple, like in maize, we can simulate a really rapid decline of leaf expansion at one point. For some other species like wheat, it's a bit more complicated, and we have to use profiled with then more than one parameter. Response to the environment, we use the time courses. So that, for example, the time courses for 3 genotypes, so the time course of transpiration and expansion rate. What you can see is that, I don't have pointers. So at the beginning, so we are still during the night, the transpiration is low. And then during the day, the transpiration increase with the evaporative demand. And at the same time, the expansion rate decrease. So this difference, the decrease in expansion rate due to the evaporative demand, we can analyze it and get the response of leaf expansion rate to VPD. So the same for soil water potential. If we combine different experiments or different conditions of soil water, we can get the response of leaf expansion rate. Oh yeah, thank you. The response of leaf expansion rate to soil water potential. I think really interesting is when we compare the response to soil water potential, the response to VPD, we obtain a correlation between these two responses. And that was the case in different species. Here it's for rice, but we observed that for maize and also probably in wheat. That means that we can use only a single parameter to simulate the response to soil water potential and the evaporative demand. That's the kind of genetic variability that we have measured in the platform. So that's for the maximum expansion rate in 5 species with dozens of genotypes and even hundreds of genotypes for maize. And that's the genetic variability we have observed for the response to the soil water potential. Remember that it's something which is also correlated to the response to the VPD. And because we have measured that in hundreds of genotypes in maize and also in mapping population, we have QTLs of sensitivities to soil water potential and QTLs of maximum leaf expansion rate in maize. I will go really rapidly on that, but we really often forgot about leaf weeds. If you go to Google Scholar or Easy Web of Knowledge, you will see that there's not a lot of paper speaking about leaf weeds. It's something like 100 times less than compared to leaf expansion, leaf elongation. And we adopted the same strategy with model of development, model for the maximum leaf widening and the response to the environment. Just an example. So here is the increase in width and length for a single leaf. And you can see that the timing of elongation and widening are different, with the widening occurring well before leaf elongation. So first, we have to consider that in the model of development. Well, you can imagine easily that maximum widening rate will be really different from leaf elongation rate. And more surprising was the response to the environment. Those responses were really different compared to the responses of leaf elongation. And especially, we had the major effect of the environment was radiation. And we had good correlation between leaf width and the intercepted radiation. And that was valid in the field, in the platform, etc. So for the efficiency of light capture and carbon assimilation, we use another phenotyping platform. This platform is the PhenoArc platform, the same one which was used by Odd in the talk this morning. So we can measure 1,600 plants. The plants are moving on conveyors, and they are imaged in an imaging station from 1 to 4 times a day, with a picture from the top and 13 pictures in total from left, from different angle. And then we can follow the growth of different plants, genotypes, etc., all over the crop cycle. So that's, for example, the increase of leaf area for in a panel of maize hybrids. So we had a really big variability in leaf expansion. And from this experiment, we can again calculate the response to soil water potential, doing that for many, many hybrids and perform genetic analysis. Then we have we have QTLs for leaf expansion rate. That's fine, that's stabilized. So we have QTLs for leaf expansion rate. We have QTLs for the sensitivities of leaf expansion rate to soil water potential and the evaporative demand. And we have also QTLs for transpiration and transpiration efficiency. So now if we want to speak about carbon assimilation, so I think you know, all of you, this equation, Monti's equation. Biomass can be, is the integral of the intercepted radiation and by the radiation use efficiency. And the intercepted radiation is therefore the quantity of light on the plant and the percentage of interception. The problem in a platform is that it's not a uniform canopy, and each plant has a neighbor, and this neighbor is from a different genotypes, or it could even be from a different species. Then we have to calculate for each plant the intercepted light in the platform. So I go rapidly on that. You had some poster on that. If you want to have, if you have more questions, Lorenz Cabrera is here, and he will explain you how in details it's calculated. So I go rapidly on that, but what is important is with 3D models, they can simulate for each small square, each 3D volume, the density, the leaf area density, and the angle of the different leaves. And so they have the plant architecture. They can have the biovolume, which can be transferred to biomass. And they have also models for calculating the local available light in the platform. So at each position in the platform and at each day during an experiment, they know what was the available light. So with these two processes, they get the biomass and the intercepted light by the plant. If you look at this, here it's two experiments for the same genotypes, so that's the increase of biomass with time. You see that their time course are different, but if you express the biomass as a function of the intercepted radiation by the plants, you obtain a unique relationship, and the slope of this relationship is by definition the radiation use efficiency. And so this radiation use efficiency is heritable, is robust, and we can perform genetic analysis. And I go really rapidly on that too. To calculate the light interception efficiency in the field, they can calculate a 3D virtual canopy and calculate what will be the interception of this plant in the field. And they have performed genetic analysis on all of these components, so like the light interception and radiation use efficiency, and then they have QTL for all of the aspects. So my conclusion is not at the end, is now. So we have fields, the phenotypic profile. Then for hundreds of genotypes, we have parameters for development, for leaf growth, for transpiration, for light interception, and for radiation use efficiency. We have also performed genetic analysis. Then we have QTLs, and then these QTLs and this phenotypic profile can be inserted to a crop model. So I will give you just a few examples. It's the beginning for us. So we inserted the parameter of development and leaf growth into a crop model. So it was based on AppSim. And we use different sets of trials for validating and simulating our results. So first, validating leaf area. So that's done with the network from the DROPS project. We have simulated length. So you see that it's not too bad with the measured length. For weeds, it's also not too bad. It's the black point. And it's really better than what was before, the gray point. And we improve the simulation of leaf area in the field. We also tested if the development, the change in the model of development has an impact. I go rapidly on that, just to say that we try to simulate the best combination of parameter of development for different situations in Europe. And what we observed was that our simulation and the observed value were not too bad. That's for sowing date and that's for heading date, so as a function of latitude. And we also tested if our simulation of yield were not far from what was observed in the field. So we simulated yields as a function of latitude for well-watered fields. That's for rain-fed fields, and the green is considering the percentage of irrigated fields. And it was not far from what we observed in different countries. And so now that was the goal of this talk, is to be able to simulate when an allele has a positive or negative effect. So for that, we consider that in each field in Europe, the parameters of development were optimal. So the farmer has adapted already the best combination of flowering time and phyllochron. And we inserted a variability on maximum leaf expansion here, and a variability on the sensitivity to soil water potential and water deficit. So I will give only one example. So it's the comparison between these two genotypes, with one genotype being not sensitive to soil water potential, continuing to grow even under severe water deficit, and another more sensitive, which stopped growth really early during drought. So that's the results, the European scale. So when the results here are red, it means that it's positive to keep going and keep growing under soil water deficit. When it's green, there's more or less no effect, and when it's blue, it's better to be sensitive to soil water potential. So you've seen that that's a genotype by environment interaction. It's something that we could have measured in the field, and it correlates well with the water deficit around flowering, more or less as expected. But what is important is we were able to simulate this effect, the G by E effect in the field. So thanks to first all colleagues for their work, their time, and their slides also. And also thanks to Graham Hammer and Greg McLean in Queensland University for their, I have no idea where they are, but for their help in AppSim modeling, etc. And thanks a lot.
Thank you. Even though we have 1 minute left, but I'm afraid we will ask questions this afternoon.